We'll be wrapping up this unit of the semester with the study of early mammalian development. This chapter of your textbook covers both mammalian and avian development, but will only be focusing on mammalian development for the sake of time. Nevertheless, there are similarities between the development of birds and mammals. Although we'll be mainly focusing on mouse embryonic development, we'll highlight differences and similarities between mouse and human development. Both birds and mammals are amniotes, and even though avian embryos develop outside the body of their mother, while mammals develop internally, both of these embryos possess an amnion, and that allows development to happen on land, since the amnion provides a source of water that prevents desiccation. This is different from amphibians, which are anamniotic vertebrates, since they lack an amnion, and as a result, they must develop in water. Mammalian development is notoriously difficult to study because it is internal, but mammalian embryos can be cultured prior to the stage of implantation. Another factor that makes the study of mammalian development difficult is that in mammals, the number of eggs that are ovulated, which is typically about 10 for mice and many animals, that's much less compared to other organisms like sea urchins and amphibians. The study of mammalian development is also complicated by the fact that the eggs are very small, whereas amphibian eggs are much larger. For example, a human oocyte is about 1,000 times smaller than an amphibian oocyte. Despite the difficulties of studying mammalian development, much has been learned from the study of mouse embryonic development. Mice are very prolific, and their embryonic development happens relatively quickly, in 19 to 21 days. They can also be housed in a relatively small space, and they reach sexual maturity between 6 to 8 weeks of age. In this lecture, we'll be focusing on the early stages of development, namely cleavage, implantation, and gastrulation. Recall that the oocyte, which is surrounded by cumulus cells, is transported from the ovaries to the oviduct by the fimbriae of the fallopian tubes, which sweep the ovaries. The cilia that are present on the fimbriae help to move the oocyte into the oviduct. And remember that these projections that you see here are not cilia. These projections are the fimbriae. They're present at the tissue level, and the individual cells of the fimbriae and the oviduct are lined with cilia. Fertilization takes place near the region of the ampulla of the oviduct, and then the fertilized egg moves into the uterus where it implants. In mammals, fertilization happens before the second meiotic phase is completed. After meiosis is completed, when the two pronuclei migrate toward each other, they don't actually fuse. Instead, each pronucleus undergoes DNA replication, and then their chromosomes condense and align along the metaphase plate. This means that the diploid zygoid nucleus is not present until the two-cell stage. The movement of the embryo to the uterus takes approximately 5 to 6 days in humans. As you can see, the embryo is undergoing cleavage as it makes its way to the uterus. It's apparent from these images that the early cell divisions in mammals are much slower than in other organisms. In fact, each cell division takes 12 to 24 hours to complete, so mammalian development is characterized by slow cleavage relative to that of other organisms. Mammalian cleavage is holoblastic. More specifically, it is rotational holoblastic cleavage. So what that means is that after the first cell division, one cell divides meridionally and the other one divides equatorially. Another difference between mammalian cleavage and the cleavage of other organisms is that mammalian cleavage is asynchronous. So the blastomeres don't all divide at the same time. So there will be points in development where the embryo will have an odd rather than an even number of cells. Another peculiarity of mammalian development is that the zygote genome is activated very early in development. In mice, the zygotic genome is activated at the zygote stage, and in humans, this happens a little later, but still relatively early at around the 8-cell stage. The activation of the zygotic genes involves changes to chromatin and to DNA methylation patterns. So that involves the removal of gamete-specific methyl groups, except for those that correspond to imprinted genes. Although the zygotic genome is activated early, maternal proteins are present throughout most of the cleavage stage, and they're important for early development. Recall that in amphibians, the site of sperm entry determines the dorsal ventral axis. In mammals, it's not known whether the site of sperm entry is related to axis formation, but there's a study that suggests that it determines the position of the first cleavage. There's also evidence that, at least in mice, microRNAs from the sperm may be needed to initiate the first cleavage. In mammalian development, cells are initially loosely associated with each other, but they become more tightly compacted at around the 8-cell stage, and that process is known as compaction. In these images, panels C and D both show a mouse embryo at the 8-cell stage. Figure C is prior to compaction, and figure D is after the embryo has undergone compaction. Compaction involves an increase in cadherin expression, and that increase in cadherin results in increased cohesion between the cells. This stage also involves the formation of tight junctions and gap junctions between cells. As a reminder, tight junctions are seals that restrict the movement of material from one side of an epithelium to the other side. So tight junctions between the surface cells of the embryo will seal off the inside of the sphere. Gap junctions, on the other hand, act as non-specific conduits for ions and molecules that allow communication between cells. At the 16-cell stage, the embryo looks different enough that it now has a new name, the morula, which means mulberry because at this stage the embryo looks like a mulberry. The morula stage is characterized by a small group of internal cells surrounded by a larger number of external cells. The external cells are more likely to give rise to the trophectoderm, which will form the embryonic portion of the placenta, while the inner cells will form the inner cell mass of the blastocyst from which the embryo develops. Eventually, a blastocyst forms, which is composed of a trophectoderm and an inner cell mass, or ICM, with the ICM being displaced to one side of the blastocyst.